Hi everyone, this is Himanshu and we were studying about the current affairs. And the next one in the line is about the biocomputing. Now, the biocomputing is basically uses the microbiology parts as the hardware to implement the computational device. So it uses molecular biology parts as the hardware. So it acts as basically a hardware to implement the computational devices. So by following the predefined rules, like often the hard-coded into the biological system, these devices are able to process the inputs and returns the output, and thus computing the information. So this is called the biocomputing itself. We were able to understand this thing in better perspective when we study about the DNA storages. So this is called what? The DNA data storage. Now it basically means that we will use the DNA to store the data as an alternate data storage to the binary data storage. Just imagine you are storing the data into your DNA rather than the hard disk or pen drives, etc. So this is what the DNA data storage actually means. So encoding and the decoding of the binary data to and from the synthesized strands of the DNA. This is what's called the DNA data storage. So it is basically an alternative to the hard drives and storage systems, and in the form of a DNA data storages. As we have said that the DNA consists of a long chains of the nucleotide, for example, like as we have discussed the AT and the CG, adenine, thymine, cytosine and the guanine. So these are basically act as a life information storage material. Life information storage material. And data can be stored in the sequences of these letters, turning DNA into the form of information technology. And more importantly, it is already routinely sequenced, the read and synthesized, that is written to and accurately copied with ease. And currently 16 GB of text from the Wikipedia has been encoded to the synthetic DNA, and that's really amazing. The same concept is basically for the DOT. Now DOT is basically what stands for the DNA of things. Now the DOT actually encodes digital data into DNA molecules, which then embedded into objects. So embedded. So the digital data will be encoded to the DNA molecules and which further embedded into the objects. This is called the DOT. While the Internet of Things is a system of interconnected between the computing devices, the DOT creates an objects which are actually independent storage objects. So this is mainly about the DOT. The next one that we're gonna study is the National Genomic Grid. Now, it will collect all the samples from the cancer patients through the network of the Pan-India collection centers by bringing the cancer treatment institution on the board. So the National Genomic is basically to collect, samples from cancer patients. And this research is carried out through the technique which is what we call as a genomic sequencing, as we have discussed earlier. Sequencing. And the NGG, the National Genomic Grid, will help to study the genomic factors which actually influencing the cancer and identifying the right treatment for the same, especially for the Indian population. The grid is also to be formed in the line with the National Cancer Tissue Biobank, which is also in the roadmap of the NGG. So that is let me write with a different pen. So it is National Cancer Tissue Biobank. That is what we call as an NCTB, and it will be set up at the Indian Institute of Technology Madras, IIT Madras. So at IIT Madras. And the NCTB, that is a National Cancer Tissue Biobank, it is a joint initiative of Department of Science and the IIT Madras. So that is a point that you can remember: Department of Biotechnology and IIT Madras. I think, Department of Science and Technology, not the Biotechnology. So because I often get confused, but make sure you don't get confused between these things, because this can lose marks in prelims. And this is mainly about the NGG. The next thing that we're gonna discuss is about the India's brain map. India's the India's brain map. The National Brain Research Center, that is NBRC, National Brain Research Center, is preparing an Indian brain template. Preparing an Indian brain template. Now, it is one of its kind, a database, of the brain template. So pretty much like a database, you can remember. And the brain templates are the MRI images that provide the anatomical informations of the human brain. So obviously, in name itself suggests the Indian brain map will contain the anatomical images of the brain so that we can study them. And this whole everything is under the Department of Science and Technology, which is managing everything. And its application will be same like analyzing the contribution of aging and the degeneration of the brain, Alzheimer's diseases, diagnosis, treatment, development of medicine, etc., etc. And you are pretty good in writing all these examples, right? And so yes, that's about the Indian brain map. The next one that we're gonna discuss will be about the Earth Biogenome project. I think this space will be enough. So Earth Biogenome project. Now, the international collaboration to sequence and digitize the genomes of every eukaryotic biodiversity on Earth over a period of 10 years. So this is what the Earth Biogenome is. It's just an international collaboration, basically, which will sequence all the genomes plus it will also digitize them of eukaryotic biodiversity, as there are two of them, eukaryotic and the prokaryotic. I have discussed in the environment section. And these are the one on the Earth over a period of the 10 years duration. So it is basically an open source DNA database. You can say it here like it is an open source DNA database. So its application can be again like the environmental conservation initiatives, and but the only problem here is that it may lead to the digital biopiracy, because it will be an open source. If something is an open source, means it can be used by anyone, and because of that, the biopiracy is one of concern. And because we are also aligned to the Nagoya Protocol, so it will go against the Nagoya Protocol to the Convention of the Biodiversity, which requires the sharing of benefits with the local communities. So it goes against that also. But let's see. This is what basically about the Earth Biogenome project. And I forgot the H here. Yes, Earth Biogenome project. The next one that we're gonna talk about is in vitro fertilization. That's what we call as an IVF, and the frozen embryos. I think you must know these things, because these days our Bollywood are also creating more and more pictures of the IVF. So basically what happened in case of a sexual reproduction, The beginning of the development of the offspring is fertilization of the male and the female gametes. It's called, the zygote. So if a male and the female gametes come together, they create what this is called as a zygote. Now, zygote is basically a fertilized egg or an embryo. Now the zygote is then implanted into the uterus. So this is create in vitro fertilization. This is what we create outside, right? And then it is implanted into the uterus, where it develops into the full-fledged organism into the various stages. So in the natural process, this takes place inside the uterus, but sometimes because of some complexities, it doesn't. Then we take it outside in the laboratories. We create a zygote there, and we implant the zygote into the uterus, where it can develop into an organism. So from then 8 weeks, the zygote is what we call as an embryo. So this is what the whole procedure of the in vitro fertilization is. Now there's a word infertility. Maybe you know that it occurs due to the various reasons. Means insufficient production of sperms or insufficient production of the oocyte, that is oocyte, or it can be due to the failure to implant the embryo into the uterus. So to correct it, we have an IVF procedure. Similarly, there is also the frozen embryos. Sometimes what happen for the future use, for example, like there are certain cancer patients, before getting to the radiation therapies or the chemotherapy, what they do, they stores their embryos in the frozen embryos, what we call them, so that they can use their these embryos in the future for the reproduction procedures. So this generally happens. That's what the frozen embryos means. And it can be also be used to get the desirable gametes also, something like that. So you're pretty much straightforward things. And the India has emerged as a surrogacy hub of the world since 2002, and there are a huge amount of the IVF that took place in India, especially after 2015. And for that, you need to have a no object certificates from the ICMR before doing anything like this. So these are the few basic things. The next is also in the news, that's called the designer babies. So I think you also know this thing because it has been so in the news in the recent times. So designer babies refers to babies that has been given as special traits through the genetic engineering. So babies who are given the genetic traits, and important word is specific, means what qualities I want in a babies, and I include those qualities in the babies through the genetic engineering. Means I'm designing my own baby, that it must be tall, it must be with the brown hair, and all the qualities I can decide, and I can put into their in mitochondria, and I can make a disruption in their genes, and I can have the babies like that. So this is done by actually the altering the genes of the egg, sperm, or the embryo. So you can write it here that this is done via altering what? Genes, egg, sperm, or the embryo. Well, exact word was actually the genes of all these. So because the gene exists in the egg as well as the sperms as well as the embryo, so we mutate them and disrupt it as per our requirements and the needs. This is called the designer babies, and actually it's unethical because we are playing with the natural selection process, and many people and the communities are against this thing. So this is what it is. And more importantly, we have a first gene edited babies also, and which we call as the LULU and NANA. LULU and NANA. These are the first gene edited babies, because in 2018 a Chinese doctor for the first time performed the gene editing on the embryonic stem cell using the CRISPR-Cas9 technology, and one of the couples subsequently gave birth to the twins. So that's what we call the LULU and NANA. So this is an example for that. Similarly, we also have the synthetic meat also these days. it has been discovered. Now, synthetic meat is basically a clean meat or lab grown meat, what we call, and used for the transgenic meat prepared ex vivo. The in vivo is what developed inside the human body, the ex vivo which was developed in the labs. So the stem cells taken out of the body of the animal are cultured into the petri dish. They give rise to the cell based cultured meat. So this is what it is. And advantage can be like the food security, the climate change, waste management, fortification of the food, reduce the carbon footprints, animal welfare, nutrition, etc., etc. Plenty of them, you can write it. Similarly, we also have the One Clean Meat project, also the first ever meat project in India. So you can also write it here like Clean Meat project. The first ever project in India, and it will be taken up by the CCMB and the National Research Center on the Meat of ICAR, CCMB and ICAR. And it is a lab culture meat can further be genetically modified, augments its nutritional content, and thus can help in the food fortification. So this is mainly about the biotechnology part in the science and technology. So this is the end of the biotechnology section, and from the next section in the science and technology, we will study about the defense technology, which is actually a very crucial one in the UPSC prelims. So let's jump to the next section. Thank you guys.